our invited speaker is Elin Teppa for this last talk. She's a postdoc at Institut National des Sciences Appliquées of Toulouse. Elin obtained her PhD in 2014 in Argentina. She then did a 2-year postdoc in the Structural Bioinformatics unit at Le Loire Institute Foundation under the direction of Cristina Marino, where her work was devoted to understanding the molecular coevolution in proteins and their impact on protein-protein interactions. Then she did another postdoc at LCQB laboratory at Sorbonne Université in Paris, where she studied coevolution in viral sequences and its application in the prediction of drug resistance mutations. Currently, she's working in the molecular modeling and design group at INSA Toulouse, studying the molecular principles that dictate the functioning and evolution of enzymes, mainly those responsible for region selectivity. So welcome, Elin, and please share your screen.
Okay. Well, thank you, Lucia, for your kind introduction. I'm going to talk about some bioinformatics tools to study coevolution on viral sequences that I developed during the last few years. First, I would like to start with the question, what is molecular coevolution? The definition says when a change in a position affects the selection pressure at another position, and this effect is reciprocal. Maybe it's easier to think about coevolution at organism level than a molecular level, where the typical example is pollinators and flowers. In those cases, pollinators can benefit from having long tongues that enable them to consume most of the nectar in deep tubular flowers. At the molecular level, in the case of protein, we found coevolution between amino acids. For instance, if we have positively and negatively charged amino acids which are interacting, they will need to change together to maintain the ionic interaction, right? So how can we detect coevolution? Well, given a multiple sequence alignment of homologous protein, we can find patterns of amino acids that change at the same time, as you can see in the 2 highlighted columns. However, covariation is not always explained as coevolution. Covariation can be interpreted as coevolution when explained physical contacts between residues or functional interactions. We will consider noise when the covariation can be explained by a shared ancestry of sequences, or stochastic noise. That means that they can covary just by chance given that alignment. So a variety of methods exist to compute coevolution, however, they cannot be applied to viral sequences. Viral sequences require specific methods because of 2 reasons. One is the high level of sequence conservation, which makes it more difficult to distinguish between signal and noise. And the second characteristic is the abundance of overlapped open-ready frame. That is when 2 or more proteins are coded for by the same nucleotide sequence. In those cases, we cannot be sure if the detected signal comes from protein 1, for protein 2, or for both of them. It's a frequent problem because the genomes of most viruses have overlapped genes. So I will explain a new method to detect coevolution using as a case study the hepatitis B virus. This is a good example because all proteins are overlapped with some other, and the overlapping genes comprise mostly the 50% of the entire HBV genome. In particular, we are interested in 2 proteins, the polymerase and the surface proteins, because both of them are treatment targets. A fragment of the surface proteins is used to produce the vaccine that produce the patient to elucidate the antibodies that then will recognize the surface protein of the virus. On the other hand, if a person is infected, there is a treatment with antiviral drugs which target the polymerase. This procedure, the vaccine and antiviral treatment, apply selective pressure on the virus in infected individuals, leading to the generation and accumulation of mutation in both genes because, they are overlapped. Well, despite this efficient vaccine that exists, Since 1982, HPV infection remains a major health problem because of the vertical transmission is the major route of hepatitis B transmission in countries where the infection is endemic. Most individuals with chronic hepatitis B don't have any symptoms. They don't feel sick and they can remain symptom-free for years. And during all this time, the person can spread the infection to others. And when the first symptoms appear, they can be a sign of advanced liver disease. There is also a relation between the risk of developing a chronic hepatitis B and the age. The younger a person is when they are first infected, the greater the risk of developing a chronic hepatitis B infection can be. As a result, we have young people which will need lifelong treatment, and that carries a risk of developing treatment resistance later on because of their constant use of the antivirals. And there are two types of mutation associated with treatment failure. First is the primary mutation, which is directly responsible for drug resistance and the reduction of viral replication efficiency. For example, it could be 100-fold less efficient than the wild-type virus. And the second type is the compensatory mutation, which improves the replication fitness, meaning that it restores the protein's ability to polymerize. So this kind of primary and compensatory mutation can be detected by coevolution methods. A common pair of drug-resistant mutation in this case is leucine changing to methionine at 180 and methionine to valine at the position 204. To give you an idea also of the conservation level of this protein, the reverse transcriptase domain have 84% of position with the conservation equal or greater than 99%. So the goal of our study is to identify new drug-resistant mutation, because so far all the resistant mutations were reported at the reverse transcriptase domain of the polymerase, when in fact the polymerase have 4 domains: the terminal protein, spacer, the reverse transcriptase, and the RNase H domain. And all of them interact. So our idea is detect positions that co-vary with mutation responsible for drug resistance, then they may also be involved in drug resistance. So we start working with a phylogenetic tree in the first step. The tree is split in each internal node. In that way, we generate all the possible subtree, and then the sequence corresponding to each subtree can be taken as the initial alignment and used to predict the coevolving position. And this is the strategy that we use, is apply a method that is called WIS2 in this iterative way. The pipeline is organized in several consecutive steps. First, we split the phylogenetic tree, and the resulting alignment is used as input. Then detect the correlated mutation among each pair of position in the alignment and associate a coevolution score to the pair by a color scale going from blue with correlation to dark red, strong correlation. Then a symmetric coevolution score matrix is built, and in the step number 3, we apply a clusterization algorithm to identify group of positions displaying the same pattern of coevolution score. Here you can see represented by the same color pattern. You can see 2 clusters, one composed by 2 positions and the other by 3 positions. Then we iterate the step 1 to 4, and the iteration ends when all the possible subtree are considered. And at the end, we include a step of a reduction of redundancy cluster generated by this iterative approach. And we consider 3 possible scenarios. In the first one, its 2 clusters are identical. That means that it's composed by the same position. And the 3 are not disjoint. Only the cluster with the best p-value score is considered. If 2 clusters are identical and the 3 are disjoint, both clusters are retained. In this case, the iteration process allows to find 2 subtrees supporting the same coevolution between the same position. And in the last case, if two clusters are contained one in another and their associated tree are not disjoint, only the cluster with the largest number of elements is retained. That allows us to maximize the number of coevolving positions. Then we validate our method in simulated data. We use a tool that is called IMCA-Evolve, and we use tree alignment as seeds to simulate the data. These seeds have different length. And by design, we choose to have the 50% of position as covariant pairs. I show you at the top, a plot is just an example of one simulated alignment showing the entropy of each position. And the coevolving position are indicated with a dot. A yellow dot is coevolving with an orange dot. And the plots show that the pair of coevolving positions are distributed in the alignment with different entropy values. And in the table, I show you the predicted performance of the method in terms of RUC. Is indicated the median and the median absolute deviation. I think we have a good prediction considering the very high levels of sequence identity. I show you here the result for the polymerase, where here each row represents a cluster of coevolving positions. The positions in red are the known drug-resistant mutation, and all the circles are the new detected coevolving position. I highlighted one cluster in the middle. You can see the coevolving position, and at the end, the information of the subtree where this pattern was found. In this example, the subtree was composed by 8 sequences with the wild-type amino acid and 25 sequences with the resistant mutations. We also provide a p-value of the pattern, that is just the probability to find this pattern only by chance given this particular sequence alignment. So to summarize the result of the polymerase analysis, we found the detection we were able to detect positions that coevolve with non-resistant mutation. We provide a measure of coevolution between non-primary and compensatory mutation. We were able also to generate a hypothesis about the mechanism of emergence of some mutational patterns that mean which position needs to coevolve before and then a third position needs to mutate, just to generate the pattern. And we report in total new 37 mutation inside and outside the retrotranscripted domain that may impact the drug resistance. And the most important for me is that we generate a general approach to the coevolution that can be used in large set of conserved sequences. And this work was published this year. So going back to the problem of overlapping ARF, we detect coevolution in one protein, but we know that it's overlapped with another one. So the question is, do the other protein coevolve on the same position or not? And this is important in particular in the case of hepatitis B, because we have the polymerase that have the drug-resistant mutation and the surface protein that's used to generate the vaccine. So mutation inside the surface protein may generate escape of vaccine-induced immunity, escaping for immunoglobulin therapy, also cause diagnostic problems due to false negative results in serological tests, just because the antibody cannot recognize the mutated protein. As we wanted to develop a general tool, we consider all the type of overlaps. They could be on the same strand. You can see here a mutation in one nucleotide affect both proteins at the same time. And they could be an opposite strand with a frame sheet or opposite strand in phase. And we consider that two positions are equivalent if they share at least one nucleotide. So how is the coevolution in the overlapping region? Here I show you the different effect of 4 coevolving positions in the overlapping region at 2 proteins, protein 1 and protein 2. Looking at protein 1, we have 2 sequences with which maintain the wild-type residues, shown in red circles, and 3 sequences show mutation in all positions, the orange circles. A mutation in protein 1 may be coupled by synonymous substitution in the protein 2 that is shown in the first column, or by one non-synonymous substitution, like in the second column. Could be by accompanied by 2 non-synonymous substitution in adjacent position, such as in the column 3. This is because of mutation at different nucleotide of the same triplet. Or could be coupled with a variety of non-synonymous substitution, as is shown in the column 4. And the cluster of coevolving position may also contain position outside the overlapping region. And at the right, you can see a representation of the results. The protein 1 shows coevolution signal between the first 4 positions. This is shown with the gray lines, and which partially coincides with the coevolution detected in protein 2. So we developed a general pipeline which takes an input, an alignment of nucleotide sequence, could be DNA or RNA sequence, and the start and end position of the overlapping orb. Then we build the protein alignment and build the trees and start the detection of the coevolving position. And the results include the coevolution of the protein 1, the coevolution of the protein 2 in a separate way, and also the coevolution of the overlapping region. So the user can analyze if there is what we call mirrored coevolution between the 2 proteins or not. And in both cases, they can see the substitution at the nucleotide level that generate the signal. Here I show you an example. This is a real result where we found a cluster of 4 coevolving positions at the polymerase with the equivalent position at the surface protein. In this case, the mutation at the polymerase confers drug resistance, and mutation at the surface allows the virus to evade the immune system. And at the right, you can see the subtree where this pattern was found. And it should be clear that the pattern was found in exactly the same set of sequences. So this is the web server that we published this year. We are happy because so far they have been used in different parts of the world. I hope this server can help to analyze viral sequences. And I would like to end this presentation by thanking my colleagues at the LCQV lab who helped me to conduct this study, the organizer of this event, and all of you for your attention. That's all.
Thank you very much, Elin, for your talk. I don't see, yes. So Yesid Cuesta-Arauz has a question. I'll read it out for you. Yesid asks, are the positions that co-evolve in the functional domains, do the amino acids that co-evolve have the same physicochemical properties? Is there any relationship regarding these properties in co-evolving amino acids? And is there any bias about co-evolving amino acids and protein-protein interactions? That's a few questions for you.
Yeah, in this case, I don't use coevolution to predict protein-protein interaction, but it can be used. There is no correlation of the type of amino acid that we found and the coevolution. Which other question was?
If the positions that co-evolve are in the functional domains?
It depends. As you can see here, we have 4 domains. The most important domain for the function is considered the retrotranscriptive domain of the polymerase. And in principle, in this case, for example, the spacer is not considered very important. However, we found coevolution of amino acid at the spacer region. So I would say no, but they could be important for interaction between the 2 domains. But as we don't know the structure of the polymerase, we don't know if they could be important or involving in protein interaction.
Okay. Let me see if there was anything else here. And then the other question was if the amino acids that co-evolved had the same physicochemical properties.
No, they should be answered with the large scale analysis. The problem with one of the problems with coevolution that we can predict them, but we don't have a way to be sure that they really co-evolve. The only proxy that we use is to use contact distance to be sure that the prediction have sense. But I would say no, there is no correlation between the physical chemical characteristics of amino acid and coevolution.
Thanks. There's another question from Erika Sousa. I hope I will read it right. It's in Portuguese. So I think the question is, are there other programs where you can do these types of evolutionary analysis without sequences being overlapped? I guess the question is whether there's other programs where you can do the analysis when the sequences are not overlapping, or I'm not sure if it's that, or whether you can still do analysis of sequences that are non-overlapping in these programs.
Okay, this method can be applied when the sequences are not overlapped. Also, the web server in the help page, they explain how to do it. And the program can also be downloaded, and you can run it locally and use in each protein that you want. It's not necessary to use the web server, and you can use it only in one protein. Besides this method, there are many other methods to compute coevolution, but not specifically in viral sequences. I just can write down a very long list of them, like EV false, for EV capping analysis, Gremlin, mutual information, DCA, and many of them have servers.
Okay, so I have a couple of questions while we still have a little bit of time in case somebody else wants to ask, but I'll go ahead with a few questions. So these types, so the results that you presented were a very detailed analysis of some specific viral proteins. Do you think, are your methods, or would you aim to scale this analysis up to many, so all the ORFs in a certain virus, or many viruses, just to see general trends? For example, I was wondering what relationship there could be between these coevolution patterns and immunogenic regions in the proteins, for example, and things like this. Then one could glean if one studies many sequences, or many proteins. Do you think this is possible, or do you really need to do these dedicated analysis on each protein to make conclusions?
Well, I know that the same method, these two methods, was applied to the entire genome of hepatitis C virus, and they were able to reconstruct the interactive network, protein-protein interactive network of the virus. So yeah, the idea, it would be very useful to apply this method to full-length genome, for instance. But then to apply a large scale to a different type of viruses, I don't think that we are on that point. The idea of generate these general approaches could be applied to different viruses, but I think then you should be careful when you analyze the results. I think it's better to be sure that what you're looking for, because there are many reasons for which to position may evolve.
Definitely. And when you make your trees, are these trees based on the protein sequences that you're studying, or are they general trees for the viral phylogeny? How does this work when you do the coevolution?
Yes, well, the user give us as input the sequence, and we perform the tree. In some methods, we also give the option to the user to provide their own tree if they want to, but we can generate it automatically.
Great. And I guess one last question that I had from my personal interest in viral proteins, as you might know. So in this analysis, there's a lot of viral families for which, of course, there's a lot of sequences because we have lots of variants, but these variants are very similar to each other. So they have very, very little divergence. So the types of of proteins and viruses that you've been studying, how many clusters of what divergence do you need to run these tests? Because in many viral families, we don't have a lot of clusters actually, so.
Yes, that is exactly why I want to show you here. In this case, we test with 96% of sequence identity.
That's great. So
it's very highly conserved. Yes.
So you can get coevolution signals even with a very high percent identity.
Yes.
So that would also allow you, for example, to study coevolution when you do in vitro evolution experiments with viruses, where you have some fast variation in some positions, but it doesn't change overall that much, I guess, the protein.
Yes, I think we can apply this kind of method. That's why we developed this method. And this is the problem of viral sequences. They are very conserved. And that's why we cannot apply all the other coevolution methods.
Great. So I think, let me see, I think we don't have other open questions. Anybody correct me from the organizers? Okay, Cristina Marina has a question. I guess, Chris, you can unmute yourself.
Thank you, Elin, for such a nice talk. And I would like to congratulate you because you solved a problem we thought it cannot be solved, which is a measure covariation between sequences which are highly identical. Mine, and also the part where you see the coevolution between different proteins overlapping is so nice, that work. So my question, it might be very technical. In the MSA you've shown, there are sequences 100% identical. Do you cluster them?
In my MCA, you say?
Yeah, the one that was in the presentation, but anyway, the question is there. Do you cluster sequences 100% identical?
No. No, we didn't apply any cluster, but if we apply a cluster, I think you refer to this slide.
No, the green MSA.
That was just an example to show the variability. No, we don't apply any cluster, but for instance, if we apply a cluster at 62% of identity, as we usually do with mutual information, we will end up with just one cluster.
No, but at 100% identity only.
No, we didn't do that. No.
But if you put just 1,000 of protein 100% identical, it might be an artifact.
Yeah, I understand that it not provides any information if you have exactly the same sequence. But no, what we did is we used a complete database of hepatitis B, and we analyzed all of them. We just take care to don't have the same sequence of the same patient. We check that with the accession numbers, but that's all.
Okay, thank you.
You're welcome.
And Elin, I guess one final question. Still, you don't need that much divergence in your sequence set, so you can work with high similarity. But do you have a minimum number of sequences that you need to apply the analysis to in order to have significance here?
Sure. We test different number of sequences. We need at least 20 sequences. That's not a lot.
No.
Yes.
Great. Very nice work, Elin. Thank you very much. And I think with this, we are finishing the session for this afternoon. So I guess I'll turn to the organizers to close on this day. Thank you very much to all the speakers and to all the attendees for their questions. It was a great session.